we are now looking at question number 31 from GATE Biotechnology 2012 paper. So the question is, match the terms in group 1 with a ploidy in group 2. So P disome, Q monosome, and R nullysome, and S trisome. So we have to match this with the correct answer, which is like 2n plus 1, 2n minus 1, n minus 1, and n plus 1. So to understand the question, let's look about chromosomal abnormalities. So there are three major types of chromosomal abnormalities, like chromosomal rearrangement. So this involves a lot of changes in the structure of the chromosome, like deletion of the chromosome segments or duplication, which leads to the duplication of chromosomal segments as shown in this figure here. And we could also have insertions or translocations, that is exchange of segments from two different chromosomes. So in this case, only the structure of the chromosome differs while the number of chromosomes in the genome remain the same. So another condition is aneuploidy. So here this results in the change in the number of the chromosomes. So an aneuploid individual might have more or less numbers of chromosomes. And there's another case called polyploidy, which results in more than two sets of chromosomes. So in this case, in the example shown here, the individual has a triploid genome, that is 3n, instead of a diploid genome, which is 2n. So aneuploidy could occur due to several reasons, like deletion of the centromere during cell division and due to translocation events, and because of non-disjunction during meiosis and mitosis that leads to the improper formation of gametes. There are different types of aneuploidy. So one is nullisomy, which is the loss of both members of a homologous pair of chromosomes. For example, in the diploid human genome, we have 2n chromosomes, that is like 48 chromosomes. We have chromosomes 1 to 22 and chromosome X and Y. For example, in case of nullisomy, we lose a pair of chromosomes. Let's say we lose both the pairs of, we just lose both the copies of chromosome 1 from the genome. So we would have 2n minus 2. So we'd have all the other chromosomes except for both the copies of chromosome 1. So the next one is monosomy. So this is the loss of a single chromosome. So in the previous example, 2 copies of chromosome 1 was lost. Whereas in case of monosomy, only 1 copy of chromosome 1 is lost. So it is represented by 2n minus 1. So we'd have all the other chromosomes in pairs except for chromosome 1, which we have just 1 copy. And trisomy is the gain of a single chromosome. So like continuing with this example, we would have 2n plus 1. We would have 3 copies of chromosome 1. So one well-known example for trisomy is Klinefelter syndrome, where the male is born with an extra X chromosome. So they would have X, X and Y instead of X and Y. So another type of aneuploidy is tetrasomy, which is the gain of 2 homologous chromosomes, and denoted by 2n plus 2. So in this case, we would have like 4 copies of chromosome 1, for example. So we would have all the normal sets of chromosome plus an additional 2 copies of chromosome 1. So hence 2n plus 2. And then there's uniparental disomy, in which both the members of a chromosome pair are inherited from one parent. So like we saw earlier, aneuploids can be produced through non-disjunction in meiosis and mitosis. So the gametes that result from meiosis with a parent with abnormal chromosomes with non-disjunction combine with a normal gamete with a blue chromosome from another parent. So this results from normal meiosis to produce the zygotes. So for example here, non-disjunction happens and we have 2 products of meiosis 1, and this segregates like these. So we have 2 copies and we have 2 copies of one chromosome instead of having just one of the red copies of the chromosome. And the other case is where no copies are found from this cell. So when this one fertilizes with a normal gamete, so the zygotes would be trisomic, that is 2n plus 1. So we would have 2 copies from the red parent, and we would have one copy from the blue parent. So in case the normal gamete fertilizes with this particular gamete from the red parent, monosomic zygotes are formed. That is, no chromosomes from the red parent and just one copy of the chromosome from the normal parent. So this is an overview of how aneuploidy is formed. So in the earlier example, we saw non-disjunction in meiosis 1, so which led to two copies of this in one of the cells and no copy in the other cell. So in this example, the non-disjunction happens during meiosis 2, while meiosis 1 forms normal cells. So in this case, we'd have one daughter cell with both the copies of from the parent red and the other one with no copies. So when these fertilize with a normal gamete, we find trisomic and monosomic zygotes. So whereas the other one with one copy from each of one copy of the red chromosome from the red parent in each cell fertilizes with the normal gamete, it leads to the formation of normal diploid zygotes. And now just to brush up the concept, so let's say a diploid organism has 2n is equal to 48 chromosomes. So how many chromosomes will be found in a trisomic member of this species? So we learned that trisomy is a condition where the total number of chromosomes is represented by 2n plus 1. So in this case 2n is 48. So 2n plus 1 would be 49. So the trisomic individual would be having 49 chromosomes. So here's an example of the effects of aneuploidy in plants. So mutant capsules in Jimson weed, that is Datura stramonium, result from different trisomies. So each type of capsule is a phenotype that is trisomic for a different chromosome. So the structure of the seed case in the wild type with 2n chromosomes would look like this, whereas the trisomic seeds which has trisomy in different chromosomes look like any of these phenotypes. So they're either rolled or glossy or elongated or like a globe, and all many different phenotypes. So aneuploidy in humans can lead to several abnormalities. For example, in case of sex chromosome aneuploids, one example of a condition is Turner syndrome, where the female is born with only one copy of the X chromosome. So this is the loss of a single chromosome, and so it is monosomy, which is 2n minus 1. So in case of Klinefelter syndrome, so this is where a male is born with an extra X chromosome. So it is 2n plus 1, and this is an example of a trisomy. So aneuploidy in the autosomes can also lead to diseases. For example, trisomy in the chromosome 21 of humans leads to a condition called Down syndrome. So primary Down syndrome is where 75% random non-disjunction happens during the egg formation, whereas familial Down syndrome is where Robertsonian translocation happens between chromosomes 14 and 21. So primary Down syndrome is caused by the presence of 3 copies of chromosome 1. So this is a karyotype of a person who has primary Down syndrome, and you can see that all the other chromosomes are normal, whereas you can see 3 copies of chromosome 21. So whereas in case of familial Down syndrome, the translocation of chromosome 21 onto another chromosome has occurred here. So here you can only see the short arm of chromosome 21. So the long arm of chromosome 21 is attached to chromosome 15. So this karyotype is from a translocation carrier who is phenotypically normal but is at increased risk for producing children with Down syndrome. Other examples of autosomal aneuploids are trisomy of chromosome 18 that causes Edwards syndrome, and trisomy in chromosome 13 which causes Patau syndrome, and trisomy in chromosome 8. It is also usually seen that there is a correlation between maternal age and giving birth to a child with autosomal aneuploidy. So this plot here is an example of older the maternal age, higher the cases of Down syndrome. So there is a correlation between maternal age and the incidence of giving birth to such aneuploid children. Uniparental disomy is another term in aneuploidy. So this is an abnormality where both chromosomes are inherited from the same parent. So that would be represented as n plus 1. So the haploid genome would, like, it would just have n plus 1, which comes from the same parent as denoted as two red chromosomes over here. So this is an example of uniparental disomy. So there's no chromosome from the other parent. So we have so far seen about chromosomal rearrangements and aneuploidy. So another form of chromosomal abnormality is polyploidy. So polyploidy is the presence of more than two sets of chromosomes. So it could be of two types, autopolyploidy or allopolyploidy. So autopolyploidy can arise through non-disjunction in mitosis and meiosis, and this occurs when organisms have more than two sets of chromosomes from the same species. So that is, like, in general, like for example in humans we have 2n chromosomes. So polyploidy would result in 3n or 4n chromosomes. So autopolyploids can originate from issues that arise during mitosis, such as doubling of the chromosomes without any cell division. So this would result in a tetraploid organism, that is 4n, from a diploid organism. So the other one is allopolyploidy. So this occurs when an organism has more than two sets of chromosomes from different species. So an example of allopolyploidy is Gossypium, a species of cotton, which are hybrids formed from two diploid species of cotton. So, in conclusion, chromosomal variations play an important role in evolution, giving rise to new functionalities and even new species. So, now coming back to the question, so disome would be n plus 1, and monosome is the loss of a single chromosome, so it would be 2n minus 1, and nullysome